Well,
Right, good morning. What we're going to have a look at today is this. We're going to look through biology, the AQA syllabus. We're going to go and make our way through this topic by topic. So our first topic is all about cell structure and transport, and we're going to have a look at this. So let's get in and have a look at the idea of what we've got. So the first part of this is to go and have a look at the world of the microscope. I'm not going to bore you by getting out a microscope. Most of you have probably seen one, and I've got some other videos on that we can have a look at. So what I want to do is take a look at what we would see down a light microscope. And looking down a light microscope, we're going to see things like this. This is a slide I made of a pineapple, and we can see the different types of cells in here. This isn't on particularly high power, but we can see the xylem cells, we can see the phloem cells in this vascular bundle in a leaf of a piece of pineapple. We can also have a look at one of the favorite experiments, I suppose, which is look at a cheek cell. And this cheek cell of one of my students, and having taken a swab, put it in a dye, methylene blue, for a little while, then we've looked under reasonably high power here. It's about 800 times magnification, and this is one of my students' cheek cells, and we can see going around here one cell, and there's several cells here. There's another one there. We can see there is the nucleus, but we really can't see inside this cell in any detail, and this is the problem with the light microscope. The light microscope simply doesn't allow us to see things well enough. If we want to see any better, then we need to go to an electron microscope. But these are a little bit expensive, probably more the cost of my house rather than the cost of something I could afford to buy easily. The formula that we come up with is image size and magnification and size of real objects. So magnification is the image size over the size of the real object. And usually in an exam, we'll come across a question like this, and it's fairly common to do so. Now, our next thing we need to know is all about plants and animal cells. And there are some really nice pictures of these sort of things you'll see on the internet. But what we need to be able to do is have a look at these better ourselves. And let's have a look at an animal cell. So let's draw ourselves an animal cell. And we're going to draw ourselves a plant cell. Now, plant cells tend to be more regular shape than some of the animal cells. Tend to means that it is true in a few cases, but not in all. And the first thing I can see that is a major difference between a plant cell and an animal cell is that we've got this cell wall. Both have this cell membrane, but the plant in addition has a cell wall, and this is made of a substance called cellulose. Cellulose is the stuff that we can't basically eat, and well, we can eat, I suppose, but we can't really digest it ourselves. And it's able to be digested by cows, sheep, goats, and the like, but we can't do anything with us, and that forms roughage in our food. So in each cell, be it plant or animal, we normally find that we can see a dark structure, as we saw in that picture that we looked at under the microscope a moment ago. And this is the nucleus. If we can start looking a little bit inside the cell and going up in power a little bit more, what we usually find inside a plant cell is this thing, a vacuole. Now, vacuoles do exist in plants and animals, but this one is a permanent one. In other words, the plant always has this vacuole. And under the microscope, the other thing that we can just about see are the chloroplasts. And we can't really see much about these chloroplasts apart from they're green, I suppose. That's one thing that we can see about them. And if we go back to our slide, we can see in some of these cells here the chloroplasts. If we zoom in on these a lot more, then we can just see they're basically green bags. Not very helpful. We can't really see any more under the light microscope. So these are basic differences between plant and animal cells. So we can say that our plant cell, it's got, as a difference, it's got a cell wall. It's got this permanent vacuole. And we've got chloroplasts. If we look at other similarities between the cells, we can see under an electron microscope perhaps a little bit more detail. We sometimes see coming out of a nucleus a sort of a long trailing like structure, and looking at this a bit more carefully, we can see there are dots on it, and it gives it its name. It's actually called rough endoplasmic reticulum. Can't be seen under the light microscope, but we get these bits and pieces here, and they are important to us because these little dots, these are called the ribosomes. Too small to be seen under the light microscope, but these ribosomes are the place where protein synthesis takes place. Other differences or similarities, we do find some small little items inside a cell, sometimes called organelles. And these things have got a horrible spelling, an horrible name. These are the mitochondria. And we think these were probably little bacteria that got sucked into a larger bacteria, and they became incorporated into the cells, and they provide the source of energy, of power to the cell, producing these energy chemicals that are widely used by the plants and animal cells to keep them going. And basically, for GCSE, this is all we need to know about the cell. It's all suspended in a liquid, and this liquid is called the cytoplasm. We need to remember all of those pieces to help us with our GCSE science. Right, now let's have a look at the next thing we need to have a look at. So we've got to remember these basic differences between plant and animal cells. Plants have got everything that an animal cell has got. There are three differences though. The permanent vacuole, this is filled with sap, and this is what keeps the cell of a plant stiff. You've got things like a skeleton to hold you up, whereas the plant doesn't. You know, the last time you had a look inside a plant, you didn't see a skeleton, I'm sure. And what it's got instead is water holding it up, and that's in this vacuole. And more of that later. We've got a cell wall made of cellulose, and we've got chloroplasts, these green little things found only in plants, and they're designed to provide energy to the plant. Right. Now, there are two sorts of cells, eukaryotic and prokaryotic cells, and let's have a look at these. Right, a eukaryotic cell we think of as one of those that I've just drawn. We can see that it's got a nucleus, it's got these tiny little mitochondria in it, it's got these ribosomes, either free or membrane bound. But if I look at this type of scale, then here is a prokaryotic cell. So this one here, eukaryotic. And a prokaryotic cell. Let's take this prokaryotic cell and let's make it a lot bigger. Now, inside this prokaryotic cell, we will see not a nucleus, but we see basically a strand of DNA. These are too small to have a nucleus because, well, the nucleus is actually bigger than they are. They don't have any mitochondria because the mitochondria is probably larger than these. They do have this strand of DNA, and sometimes they have other circles of DNA. These are called the plasmids. And these are not particularly nice little things. These plasmids often carry basically resistance to antibiotics, so quite useful to the plants and the bacteria, not so useful to us. Yet we can use them to help with genetic engineering, which we can do quite well. They do have some ribosomes in them, but these are actually smaller than the ones that we find in a eukaryotic cell. It's got normally some sort of cell wall, and occasionally you'll find on these bacteria, but not exclusively, some flagella. Some do have them, some don't. the idea is maybe that helps them move, but some manage without. So the jury's out on that one, really. So we've got a cell wall. Sometimes we have a slime coat, and this protects the bacteria from things like your stomach when you may eat these bacteria, and it may help them get through the stomach to move on to live in your small intestine, where they can cause either good or considerable amount of harm. So eukaryotic cells and prokaryotic cells. The major difference is size. Eukaryotic cells probably 100 to 1,000 times larger than the bacteria. Right, our next little bit to move on to then is specific types of specialist cells, and we'll find a whole load of these in animals such as you. So we look at what sort of cells are inside a human. So let's have a think about a human or animal cells. And there are about, I think it's about 216 different types of cell available in your body. So a fair number to sort of go and have a look at. And you're asked to be able to remember just some of those. Thinking about our blood, then we can think of at least a couple here. We've got our red blood cells, often called RBCs. We've got our white blood cells, which protect us from infections. So we've got our red blood cells, we've got our white blood cells. Now, other types of cells can you think of? Well, we've got nerve cells running around the body. Now, if we think about some of these cells and we have a look at some of these cells, we can see something quite interesting. If I go and have a look at some of my cells, I can see a whole load of them. And here I've got some blood cells to have a look at. Unfortunately, in my family, when you cut yourself, you don't sort of immediately put a plaster on your finger. No, you run and put some of this on a slide so we can have a look at them. And here are some red blood cells at the same magnification as that cheek cell. And you can see there's an enormous difference in size. Something else which is interesting about these red blood cells is that they actually don't have a nucleus. So in many respects, they're not cells. They're dead. They're just stuff full of one chemical called hemoglobin. So if we're looking at a specific example of a cell, then perhaps the red blood cell isn't probably one of our best choices to go for. So we've got our white blood cells, nerve cells, and we can often think of the muscle cells to move us around. Now, comparing size, red blood cells you saw were small. We had our other cells that we just saw there. We can see our cheek. They're called epithelial cells. Basically skin cells, cheek skin cells. And you saw that the cheek cells are a lot bigger than the red blood cells. But how about a nerve cell? Well, a nerve cell's quite an interesting one to have a look at, because you look at the body of this cell and you see it's quite small, but in fact, there's some of the largest cells that you can see in your body. The largest one that I possess goes from the end of my big toe all the way up my leg to the base of my spine. So quite a long cell, very, very small. And if these were the size of those cells, you can see that if you come to surgery, we have a significant problem that you cannot sort of stitch up two cells together because they're actually smaller than a needle. And the fibers of a nerve cell, the axons as they're called, are normally much, much thinner than that. Other cells that come to mind immediately come from reproduction when we think of the egg cell and the sperm cell. The egg cell is about the largest cell that can be seen in the body. It's about 1/10 of a millimeter across, but still that is, I suppose, just about visible to the naked eye. And sperm cells, again, quite small. So we've got quite a range of cells, and they look very different. So if I were to draw out something like a nerve cell, then quite often they have a peculiar shaped little body, and they've got these little connectors coming out, reaching out to some cells, and then they'll have a much longer one, perhaps even surrounded by other cells. And we have our cell that does the two things. It has a nerve ending, which might go to something like a muscle cell. So these may touch muscle cells, and up here, these may touch other nerve cells so that information can be passed from one to the other. And in there, we'd see we've got our nucleus, and we'll have our mitochondria and our ribosomes and our other things. But the structure of this cell looks very different to our cheek cell, which we had a look at a while ago. So my pen's collapsed, there we are. So nerve cell and the cheek cell are different in size, different in look, different in structure, different in function, but they all have the same things. And the same is true if we look at muscle cells, which have lots of fibers in them that contract. We've got the egg cell, which is enormous in comparison with all the others and contains all the material necessary to divide many, many times when its DNA complement is increased by a sperm. And a sperm cell, again, very small, very specialized, with a long tail and tightly packed mitochondria, and just really a nucleus containing half the amount of DNA ready to be delivered to an egg. So that's a human cell. Let's have a look and see about a plant cell. Plant cells, we've got a whole variety of different ones. And if we look at a few of these, so I'm just trying to find the slide. If we look at these cells, we can see that some of them look quite different. We've got some cells that are green here, and some cells that really don't look very green. So we've got in here two sorts of cells, and let's look at them. We've got then the xylem cells. So we have as a structure a vascular bundle. So here's our plant cells. In our vascular bundle, we have the xylem cells. Now, these are actually dead cells, a bit like red blood cells, I suppose, that are dead, but they perform a very useful function. The xylem cells form basically a tube, and the tube goes from the root to the leaf. And through this, water is passed up from the root to the leaf via a process called transpiration. The other types of cells in this vascular bundle are called the phloem. And the phloem take the glucose, the sugar, the other materials that are made inside the leaf, and they take that from the leaf and they distribute it to all the cells in the plant. What other cells can we see? Well, we saw those green cells, the ones with lots of chloroplasts in them. And if you look inside the leaf, we can see the palisade mesophyll cells. The palisade mesophyll cells are your main photosynthetic cells found in a plant. These simply do all the work of turning light energy and basically splitting water and carbon dioxide and rejoining them together to make glucose. Other cells that are famous are the root hair cells. And sometimes we have a look at those in the plant and we can see not just a little cell, and this one usually requires some sort of diagram, and it has this tiny little hair coming out, and this increases its surface area so that this plant cell can bring in lots more water and other materials to the plant. Nutrients, it's a common thing when I talk about plants, that students think that these sort of cells, they sort of supply all the food to a plant. It doesn't. It just gets hold of extra material, things in the soil that the plant might need, nitrates, phosphates, other ions like perhaps sodium, magnesium, potassium, and they're brought in along with the main thing, water, into the cell, and that is then used to be transported to perhaps other cells like the xylem. So a root hair cell just bringing in water and a few extra nutrients to the cell. So that gives us the plant cells. Our next and last thing that we're going to have a look at here is diffusion. Diffusion is the movement of a substance from a high concentration to a low concentration. And we can speed that up with simply increasing the amount of stuff we have, or by increasing the temperature. A common way of doing this is we take a glass of squash. Let's take a glass, and I'm going to fill it with water. And then very carefully, I'm going to take a tube and I'm going to push this tube down to the bottom of the glass, and I'm going to put in here some squash. And what happens is the squash tends to sort of get down and stay at the bottom. You can do this carefully yourself as an experiment, and it works quite well. Either you can use a tube, or sometimes you can actually just pour it very carefully down the side. And we end up with some water and some squash. But if we leave this for a while, we get typically just a glass of squash. Now, what's gone on? Well, if we look at just what's going on in here at the moment, so at the bottom of my glass, this is magnified an enormous amount of times, we've got some molecules of squash, and near and around them we've got some molecules of water, and you can see my squash molecules are much bigger than my water molecules. They probably are much bigger. And all the time, the molecules are moving around. Right, so let's have a look and see what happens. Well, these molecules move around. So if I take this little molecule, it's moving around, and it collides with another molecule, and it will push that molecule off in a direction. But that molecule might be moved by another molecule. And what we're going to do is we're going to have a look at a little experiment to see what happens here. So what I'm going to do is I'm going to play a little game, and the little game involves some dice. Here we are. I've got my dice, and I'm going to divide them into two. And what I'm going to do with these is we're going to make up some rules. And I'm going to have, first of all, my purple dice and my pink one. And my purple dice, it's going to depend on what number we get. We're going to go off in a particular direction. So if it's a 1, it's going to go up. 4, it's going to go down, and so on. And the pink one, it's going to go that distance in centimeters. And exactly the same for the blue dice, or the blue die, to be correct, and the green one. The green one is going to give me my distance, and the blue again is going to give me my direction to go in. So let's start, and I'm going to have my purple dice here, and a bit lower, let's put my blue die, and we'll see what happens. So I've got my little dice tower here, and we'll just drop the dice in here, and we get our particular values. So for purple, it's 3 and 6. So we're going to go direction 3, and I'm going to move 6 centimeters. The other one, we got a 1 direction and 1 for distance and 2 is my direction. So let's do this one. There. Right. Let's try this again. We've got this time a 5 on the purple. If I put it there, we can see just about 5 for the purple. So that's this direction and a distance of 3 centimeters. And the other one goes up in a direction of 1 and 3 centimeters to do this. We'll run it a couple more times to see what happens. I usually get students to do this several times. So I've got on the purple side 5 and a 5. It's pure luck that I managed to get all 5s on that. Don't think I could probably do that one again if I tried. Let's just put these in. So we've got direction 1 and going up 3, so it's reversing its direction. So it ends up there. And the other one's going direction 2. Oh, they are both going back the same direction. 5. One more time. Things don't ever work out when I try and do these things. I've got a 6 and a 1. So this one's going to go a direction of 6. 1 centimeter, and the other one's going to go a direction of 3 and for 2 centimeters. We can see from this it's called a random walk, and this is how basically molecules, atoms move. This is diffusion. It is the random movement of molecules as they move around, and they move around knocking into one another, which they didn't in this case, and as they move, our solutions, our gases all become mixed, because it moves from a high concentration where we've got lots to a low concentration where we haven't got many. And as we do so, we see everything becomes mixed and basically evenly distributed. So there we have diffusion, the movement of particles from high concentration to a low concentration. In the next video, what we'll have a go at doing is starting off looking at osmosis and active transport, and see if we can finish this topic and move on to the next. I'll see you next time. Thank you. Bye-bye.